This is the Professor, CBMD PhD, and today is Sunday, August 16, 2026. It is 5:11 and 31 seconds, p.m. Eastern Standard Time, and today's research, the mystery of bacteria. So let's hop to it here. Scientists discovered that bacteria can turn dissolved uranium into a surprising stable form, revealing a potentially powerful new tool for cleaning contaminated water. Date August 9th, 2026. Source: Helmholtz Zentrum. Deep underground bacteria may be capable of doing something remarkable, trapping toxic uranium before it spreads. When researchers fed microbes in uranium-contaminated mine water with glycerol, about 95% of the dissolved uranium disappeared within 130 days. The bacteria helped convert it into an unusually stable uranium compound that had rarely been observed before. Uranium, a radioactive metal, is typically locked inside minerals and soil. However, mining and other environmental processes can change uranium into forms that dissolve in water. Once it becomes mobile in this way, it can spread through the environment and create problems because its toxicity. Bacteria found in soil and water are essential parts of natural ecosystems, and some species are capable of processing substances that can be harmful in humans and other organisms. There are bacteria that can metabolically utilize the heavy metal uranium, which is toxic for humans. Our group of investigators had already revealed that bacteria can use uranium dissolved in water for their metabolism when they have access to glycerol as a food source. Glycerol is a basic component of plant and animal fats. It can also form naturally. For example, when fungi break down wood, the researchers wanted to determine how effectively bacteria can reduce the amount of uranium dissolved in water and identify the chemical forms created as the microbes processed the uranium. To investigate, the team collected mine water from a flooded uranium mine in the Ora Mountains. In the laboratory, they added a controlled amount of glycerol to samples of the water and kept them in an environment without oxygen. We wanted to create natural conditions for the bacterial community already existing in the mine water because, at a depth of approximately 2,000 meters, there is usually little or no oxygen in the mine. With conditions suitable for bacterial growth, the microorganisms began using glycerol as a food source. Over time, the amount of uranium remaining dissolved in the water dropped dramatically. After 130 days, only around 5% of the uranium dissolved in the water remained in the samples. We suspected that the bacteria had incorporated the uranium in their cell walls. We already knew about accumulation processes from the literature. The researchers were then able to confirm that uranium had indeed accumulated within the bacteria cell walls. The next question was exactly what type of uranium compound had formed. To find out, the researchers turned to advanced microscopy and spectroscopy. The scientists examined the bacterial membrane to determine the chemical states in which the uranium was present. Chemists use the term valency to describe how many hands an atom effectively has available to connect with other atoms in a chemical compound. Uranium usually occurs within a valency of 4 to 6. Pentavalent uranium does exist, but it is rare, or only transient. Until now, it had been seen in an unstable oxidation state. So the findings of our study were extremely surprising because, in the biomass analyzed from our experimental runs, an unusually high proportion of the uranium identified was also pentavalent uranium. The discovery was unexpected because the pentavalent state of uranium had generally been regarded as unstable and short-lived. The researchers also determined that the pentavalent uranium combined with iron and oxygen to form FeUVO4. This uranium compound doesn't have a name yet, as it is comparatively new. It was first demonstrated in a study in 2020 in which soil samples from parts of Croatia contaminated by uranium ammunition were analyzed. It was found that even under the influence of atmospheric oxygen, this uranium compound had remained stable for more than 25 years, but until now, We didn't know how this compound is formed in nature or that bacteria play a role in its formation. Additional experiments produced another surprising result. When the researchers exposed dried bacterial biomass to oxygen, the amount of FeUV04 increased rather than decreasing. That finding suggests the compound can remain stable even in the presence of oxygen, adding to evidence that bacterial activity may help convert mobile uranium into a form that is much less likely to move through water. Our study has revealed for the first time that bacteria supplied with glycerol as a carbon source can convert toxic uranium dissolved in water into a stable chemical compound. We still have to investigate to what extent bacteria might help to render uranium harmless for remediation purposes. The researchers now plan to study uranium-binding bacteria in greater detail and investigate the biochemical and geochemical processes that make the transformation possible. A better understanding of those mechanisms could eventually help determine whether bacteria can be used effectively in efforts to remediate uranium-contaminated environments. And this video was created for educational purposes only. My code name is Will. I'm Professor Christopher Brunet, leader of research, scientist ufologist, psychic computer technician, computer programmer, philanthropist, writer, mathematician, artist, photographer, film producer, chef, animal, and nature lover. My passion's world peace. Have a great day.